All right, welcome everybody to the February edition of the Biotech Talks here at UWBC. Today is a very exciting day because it's all about PacBio, and our title, or first of all, sorry, our person is Chris Black. This is Chris. Welcome, Chris. Our title is Introducing the PacBio Revio System, a game-changing tool for omics studies. And my name is Molly. I'm the lab manager for the DNA sequencing facility. We have 2 Sequel 2s currently, and I wanted to share some stats with you in case you guys really wanted to kind of know a little bit more about the quality expected from our current sequels and just knowing that it's going to get better with the Revio. So if we start up with 1.5 million reads, minimum quality is Q20. Of those reads, we get 1.2 million that are Q30 and above. So we're already, we dropped a little bit off. Of those 1.2 million, 900,000 are Q40. Of those, 710,000 are Q50. And of those, 600,000 are Q60. So if anybody doesn't know what Q60 is, that's 99.9999% accurate. So
it's pretty good.
Which is very good. And that's only what, 40% of the original reads that we started with. So we are doing pretty well at the quality over here. So please come check out some PacBio and listen to Chris talk all about PacBio.
Thanks, Molly. I appreciate it. Thanks for having me today. My name is Chris Black. I actually live here in Madison. I came to Wisconsin from California originally as a graduate student at McCardle. So I've been around for a long time, and makes me really happy to have the opportunity to speak here in Madison. That's always really fun for me. Today, I'm going to talk primarily about the Revio system, which is a brand new platform that we've just launched. In fact, we've just shipped the very first ones out of PacBio hands and into customer hands in a couple of sites around the world. And so we're just now starting to see people utilize this technology. But a lot of what I'll talk about is using the previous generation of our platforms, which is the SQL platform that Molly had just mentioned. So the way they work is very similar. We'll talk about that a little bit. But I'm really excited for what this new platform, the Revio, can bring to the folks here in Madison and across the country and around the world. We have a pretty manageable size here, both online and in person. So I would say if anybody has questions as we go along, ask questions. And if we're going to cover something again later, I'll let you know that. But let's have as much of a conversation as possible as we go along. So the first thing I want to do is talk a little bit about how our HiFi technology works, what it means, how we work with it. And so then we can start looking a little bit at the ways that the technology is applied. So I want to start here at a very, very high level. So super high level overview. What does the overall workflow look like for a HiFi run? We start with any sample. So if you want to look at viruses, vertebrates, humans, plants, anything that we can get high molecular weight nucleic acids out of, those are samples that are compatible with the general HiFi workflow. And we have some recommendations and even some products of our own that talk about how to isolate those nucleic acids. What's important is that we're getting high quality, long molecule, high molecular weight nucleic acids. And that's what you see in that second step. So we start from whatever those materials are, we isolate high molecular weight nucleic acids, and we shear those down to the desired lengths. Generally, that means something on the order of about 15 kb or so. We can have a little bit of a shoulder on the sides of that, but in general, we want to shoot for about 15 kb as our mean insert size. From there, we ligate these hairpin-shaped adapters and form what we call smart bells. So up there in the top left-hand corner, you can see the structure of a smart bell library. It's a double-stranded DNA insert with hairpin adapters on the end. And I'm going to say this a number of times. The important things about those smart bells is that they are structurally linear, but topologically circular. So they act like a relatively stable piece of double-stranded nucleic acid, but they are shaped like a circle. And you'll see why that's important as we kind of move around. From there, we can hybridize a primer to that adapter hairpin sequence, and we can have a polymerase jump on board and start integrating nucleotides and synthesizing a new strand of the template. We use a very high positivity polymerase that generates very accurate long reads. And then we load all of that complex, the polymerase and the primer and the smart bell, onto our chips. I was going to bring a chip and I forgot. Onto our chips that we call smart cells. And on those smart cells are very, very small nanowells. So nanowells so small that light cannot escape out of them. So when we have light signals that are captured down at the bottom of these smart cells, the diameter of the smart cell itself actually forbids light from escaping out and contaminating adjacent wells. So we have a way to capture signal directly on the surface of a chip inside that smart well, and we can do that millions of times. So we load those smart cell chips onto the sequencing platform itself, where the data is generated and analyzed, and we use that for making calls and so forth down the line. Okay, so let's dive in a little bit more deeply in that and talk at a slightly more molecular level what that looks like. Up in the top corner, you can see the configuration of a smart cell chip. So it's essentially a computer chip, like a CCD camera that would be in a phone or something like that. On top of that, we have the ports where we can actually perform the biochemistry itself. Where the shiny gold part is there is where the zero mode waveguides or those nanowells exist. And from there, when we load our single molecule libraries, so a couple of things I want to make sure I'm being really clear about here. So the molecules from which those smart bells are derived are single molecules. They are single insert molecules from the nucleic acid that was harvested from whatever your sample of interest was. We're not amplifying the molecules. We're not doing clonal amplification and making more of them so that we can see them. We're looking at single molecules. So we load those single molecules into the zero mode waveguides. And we can begin incorporating fluorescently labeled nucleotides inside the smart wells and on the surface of the chip. So you can see that depicted over here. We bring in phospholink nucleotides that are fluorescently labeled. As the correct nucleotide comes into the polymerase complex, we integrate that nucleotide, liberating the fluorescence molecule, and we can actually take a movie in real time, a real-time movie of integrating fluorescent signal via the enzyme complex that's on the template molecule itself. What you can see down here is that means that we get a signal that not only indicates which nucleotide has been integrated, but how long it took to integrate that nucleotide and how long it took between nucleotides. That's what we call kinetics. So we're looking at not only integration events, but also the kinetics that come along the way. That's really important for looking at epigenetic modulation directly from those molecules, which is something we're going to explore a little bit as we go through. So just keep that piece in mind as well. So we're making real-time base calls from the fluorescence profiles based on integration events of the polymerase, capturing the kinetic information at the same time. And in so doing, even though we're starting from millions of individual single molecules, we can build to very high consensus accuracy. Molly shared with you some of those incredibly high figures just a minute ago, through multiple circular passes around this structurally linear yet topologically circular input molecule. Okay, so there's going to be an animation that plays on this slide. Don't lose me the whole time, but make sure that you're watching that animation because it's really interesting and important as well. Okay, so when I say that we build towards consensus accuracy by moving multiple times around the input molecule, what that really looks like is what you're seeing here. So we start with high quality double-stranded DNA that's on the order of several kilobases long. We put on those loops that turn the whole thing into a circle, and then we allow the polymerase to move around the circle, repeatedly sequencing the same single molecule in a single well. What that does is allow us to start building up towards a consensus accuracy. Now, I want to be really clear about something. This is inherently a single molecule technology, and single molecule technologies almost always struggle with signal to noise issues. Because you only have one molecule that's generating your signal, any background signal can cause your error profiles to increase. What you see over here, all of these red marks and black marks and all the little mismatch molecule indicators are an actually pretty accurate reflection of the raw error profile of our reads. So we have a relatively high raw error profile, but by moving around the circle, oh, the animation stopped, that's a bummer. By moving around the circle multiple different times, we can eliminate those raw random errors through consensus accuracy. So what does that actually look like? So we're moving around the circle here. We have all of these raw errors. We're building towards a single consensus read that's very high accuracy. The reason that is because we're building each time on the likelihood that that one nucleotide is correct, and that one is correct, and that one is correct, and it's only in a very rare and completely random instance that any one of those nucleotides would have been called incorrectly. So the profile of how our Q scores build up to those extremely high accuracy scores overall is shown over here on the left-hand side. For our HiFi reads, anything that has not been read, any nucleotide, any position that has not been read at least 3 times won't be included in our HiFi analysis. So that'll basically just be thrown out. It means that we haven't achieved at least Q20, and we're not going to look at that at all. With most of the data analysis that's done, we throw out anything that's below Q30. Q30 is sort of an industry standard saying that one time out of 100, one time out of 1,000, you're likely to have a misincorporation or a misread event in that read. What we get with this system is the ability to have very long and very accurate reads that have very even coverage across the entire genome without systemic bias. So we're not sensitive to things like GC content or repeats or any other motifs that would be challenging. You do? Which ones? Okay. Yeah, it'd be interesting to dive into that and see if it's the data analysis or it's the raw data that's actually causing some of those errors. I would guess that it's likely to be more on the data analysis end. But yeah. Yes, it's fair. Yeah, it's a fair comment. Yeah. Oh, sure. So the comment was that that Roger has observed some systematic biases around homopolymers for Ts in particular, or homopolymer regions in general. Yeah. Okay. I'd love to dive into the data a little bit and see what we can figure out there in terms of understanding why that's happening and getting it fixed. Because of the length of these reads, our mappability is very high. So we can look at regions that are called dark regions, pseudogenes, and repeats within the genome that have evolutionary significance and start to understand what those mean in a way that's really difficult, if not impossible, for short reads. Because we're looking at single input molecules, we get allele level resolution and long range phasing across the genome. So if we see variants showing up, we can tell if they are in cis or in trans to one another. If you have a compound heterozygote that's actually knocking out a gene completely, or if there's a high likelihood that you have one functional copy and one non-functional copy of that gene within the genome. And overall, when we compare to other sequencing technologies, we actually have the highest performance for all variant classes, single nucleotide variants, indels, structural variants, all those sorts of things because of the confluence of all of these things together. Now, I'm making big, bold claims here, and Roger's already called me out on one of them, which is great. I'm not going to show a bunch of comparison data because I think that's really boring. We have all that comparison data and people have done that work. So if anybody is skeptical and wants to see what that comparison data looks like, I'm happy to follow up and provide some of that, but I'm not going to show a bunch of those slides here today. What I would like to do is talk a little bit more about the Revio system particularly, and then dive into some of the applications of the technology. So here's the Revio. It's a big black monolith of a box. It's actually really cool looking. Not that I think that means that you should buy it just because it's cool looking, but it is cool looking, so that's kind of an added bonus. On the instrument, we can run up to 4 smart cells simultaneously, and on each of those smart cells, we'll have up to 25 million ZMWs, so 25 million of those small features that allow us to do sequencing. What that means is on a single run, we can look at up to 100 million ZMWs, which is a ton of sequencing. We expect that that's going to yield about 360 gigabases of HiFi yield per run in a 24-hour sequencing time. We're still targeting about 15 to 18 KB overall read length on average, and we are still looking at methylation and directly calling 5-methyl-C in a CPG context on the instrument itself. So directly from the kinetics on the instrument, we're actually calling 5 bases rather than 4. Q-scores will continue to be just as high as they've always been, with, in all likelihood, well greater than 90% passing 230. Here's a quick comparison between the two systems, the system that the lab has now and the system that we're working on bringing in very soon. So the Sequel system has 8 million ZMWs, Revio has 25 million per chip, so a 3-fold increase in the number of features per chip. Sequel is only capable of looking at one of those chips at a time. Revio has 4 independent stages, so we can look at up to 4 chips simultaneously on one instrument. The run times for the Sequel 2 are at about 30 hours. We've been able to carve that down to 24 and keep the same high accuracy and long read lengths in the Revio system. What that overall equates to is that with a single Sequel system, you have the ability to produce about 88 human genomes worth of data per year. And with a single Revio system, we have the ability to produce 1,300 human genome equivalents per year. It's a 15-fold increase per system. So the total amount of data that can be acquired is significantly higher. Part of the way that we've done that is by parallelizing the internal mechanisms of the instrument. So the actual optics and signal acquisition platforms inside the system, there are now 4 where previously there was only 1. What that means is that we can do about 4 genomes at 30x in a single run. So you could take a set of tumor normal pairing samples, run a whole genome of normal, double up for 60x coverage of the tumor, and then still run a sample of MOS-seq on the side just because you were bored and you had the room to do so. I don't think you would actually run an experiment like that just because you're bored, but Josh thought it was funny. Pardon me? So theoretically, it depends on how much you would want to multiplex per chip. A lot of folks are trying to run 1 MOS-seq sample per chip, so you could run 4 MOS-seq samples simultaneously in a single run. One of the other things that we've tried to do is to make life a little bit easier for Molly and for the other folks that work in the lab, and consolidate down the way that you interface with the instrument. So not only have we taken what used to be 5 or 6 different things that had to be loaded on the instrument and put them all onto one single plate, we've also got rid of the need for an external nitrogen supply. Currently, we flood the smart cells with nitrogen so that there are no oxygen molecules that are getting in and breaking up all of our precious biochemistry. We don't need to do that anymore. We've redesigned the smart cells so that we don't need to flood the thing with nitrogen. And we have a mechanism where now the sequencing can be occurring down here in the guts of the Revio on those 4 independent sequencing stages, and still allow the platform itself to be open. So you can open the door and you can interface, you can move the plates around, you can put new stuff on, you can start up your next run before the first one has even finished. The overall workflow is much better. Sure.
Okay, we have a question from James, and he was asking, can the Revio reach 50x? And if so, in what cases? If not, what prevents that?
50x coverage?
Yes.
So I assume the question is, can we get to 50x coverage? Yes, it depends on the genome size and the total number of runs that you've used, or the total number of smart cells that you've used to analyze that sample. You can theoretically sequence as deep as you want. It just becomes a question of time and money. So the other thing we've done with the Revio platform is we have crammed it full of great bioinformatics and analysis tools. So we've integrated NVIDIA GPUs directly on the system. We've worked with groups at Google and others to build analysis tools directly on the system, which are leading to even higher read accuracy than what we see on the Sequel 2. You can see here what that looks like when we integrate Deep Consensus, a base calling tool that looks at making the highest quality base calls possible. And in so doing, we know that we're going to be generating a huge amount of data. We've actually found ways to compress our file sizes. So while we're making a lot more data, the data storage and data transfer needs are actually going to go down for the new system. I'm going to show a slide that Josh and Molly are going to cringe at because it's going to say $1,000 for a 30x genome. That's true for the consumables. So for the stuff that you need to use to run the instrument, it doesn't take into consideration Josh and Molly's time and overhead and all of the other things that it takes to actually run a lab. But the thing to note here is not that you should go to Josh and Molly and say, I want my $1,000 genome. What this really reflects is an overall decrease in the cost of a single run. So the consumables for a SQL run cost about $1,400 to $1,500. For Revio, it costs $900 and $995. So what I've just told you is that you're going to get 3 times more data for a single smart cell, and it's going to cost you about 2/3 of the amount for the consumables themselves. That's pretty exciting in terms of thinking about how you can utilize this really powerful instrumentation. Sorry, I know I'm going to cause you. I apologize. I just wanted to show some quick data. So this is internally derived data where we've used the Revio system to look at human genomes as well as non-human genomes. We're reporting very good statistics in terms of the overall read length, the overall yield, and you can see that the base quality is still very high here. In terms of what that looks like slightly more granularly, you can see the distribution of our read lengths. We still get some very, very long reads, but we're targeting something on the order of about 15 to 16 KB as an average. You can see that we still get very, very high read quality, including some of these reads that are almost likely unmeasurably high quality. They're higher quality than we're capable of or confident in calling. This is at the read level. And this over here is at the base level. So you can see that the individual base quality is extremely high when we use this consensus reading. I said we'd circle back to the idea of looking at the 5th base pair, the methyl C, 5-methyl C. Here is an example of a context where this really matters. PEG3 is a maternally imprinted gene. So we know that the methylation status is going to be different for that gene. If you look over here on the left, you can see that there are variants that are called in this gene, but they're not always sufficient to understand what the imprinting pattern is in and of themselves. If we look instead over here at the methylation pattern, you can see the blue is a hypomethylated state, the red is a hypermethylated state, and both the Revio and the SQL platform are calling these two samples similarly in terms of their methylation status. So we can use this information about methylation to do things like phasing to span through regions of the genome where we might not otherwise be able to see that and so forth. Yeah.
So what organisms is this tuned to right now?
So this should work for anything mammalian. We're looking specifically in terms of the calling that's occurring. Sorry, the question was what organisms this 5-methyl-C calling is tuned to right now. We're looking at anything specifically that's 5-methyl-C in a CPG context. That primarily means mammals. There are other epigenetic modulations that occur in plants and in bacteria and other non-mammalian species that are very important. We can still gather kinetic data on that, and we can likely still make those calls. In fact, I'll show one application that uses that directly. But in terms of the calling that's happening directly on the instrument itself, it's only 5-methyl-C in a CPG context. And so here, just some quick comparisons. The only comparisons that I think I'm showing at all are comparisons between SQL and Revio, and showing that the performance between the two platforms is incredibly similar, and that there are actually a few instances where the Revio is outperforming the current state of the art, which is the SQL platform. Any questions on how this thing works and why it matters that it works in the way that it does? Okay, cool. I want to switch gears a little bit, and now I'm going to share a number of examples of applications and data sets and work that's been done and published, or at least pre-printed by various groups around the world. This is what I get really excited about. I actually love genomics technology. I'm a nerd for genomics technology. I tend to chase around whatever I think is the coolest thing at any given time to try to be a part of. But the reason that I actually love all those things is because of the work that it enables people to do. So I'm going to share a few examples. This is meant to be sort of a survey and just a quick explanation of some of the work that's done. I'm not going to do a super deep dive on any one of these examples, but I'm happy to follow up with anybody and chat about these more as you see fit. So a couple of great examples of how people are utilizing HiFi technology and very high accuracy, very long reads to supplement the work that's been done historically. The first two I love to share here are examples from the Telomere to Telomere Consortium and the Human Pangenome Reference Consortium. And I'll let you guys read the comments that were shared by some of the PIs that are involved in this work, but they've been very complimentary about not only how the system works, but how the data type itself is enabling for trying to understand the overall complexity and composition of the genomes that they're interested in. So if we look specifically at the Telomere to Telomere Consortium, hands up, anybody know when we first sequenced the human genome? The whole human genome? Josh knows. When was it?
The whole human genome?
Sure.
I don't think we've sequenced the whole.
Well, these guys would disagree with you. The Telomere to Telomere Consortium would say that we first sequenced the entire human genome in May of last year. Not in 2000 when the cover of Science said that we sequenced the human genome and HG004 was first published. And what they can actually show in this paper, and over here, I understand it's really small, I apologize, I recommend taking a look at the paper. All of these black regions that are oftentimes here at centromeric regions or at telomeric regions, all of these black regions are part of the human genome that had never been fully assembled and reported on prior to this work. They tend to be very repetitive. They tend to be, like I say, largely centromeric and telomeric. What's actually really, really interesting, and again, I apologize at how small this is. If you look at some of these regions like these guys here, the red indicates that these are genic regions. So these aren't junk DNA that are just there for something structural. These are regions that have genes that are undoubtedly doing something in the genome that we care about. The authors basically say down here at the end, we used a whole bunch of technologies. We used all of the available genomic technologies, long reads, short reads, optical mapping, anything Hi-C, anything that we can think of to understand the full structure of the human genome. But the core, the basis of the current telomere to telomere assembly is a high resolution assembly string graph built directly from HiFi reads. Similarly, there's the Human Pangenome Reference Consortium. So the idea here being, rather than referring to a single genome, a single sample, a single person, when we're trying to understand the overall complexity of the genome, why don't we try to understand the full variability and the full complexity and diversity of the genome by looking at multiple different samples and assembling them all together into one pangenome that represents the full complexity of human beings. This is work that's funded by the NIH. They've funded up to 400 whole genomes that would be completely assembled and put into the pangenome itself. This is from a report that they issued last year that was sort of the first 40 or so of those genomes that they worked. And again, they're basically saying here, high base calling accuracy of HiFi is extremely important to being able to do this work and assemble the pangenome. And obviously, it's not just human genomes and pangenomes that we're interested in. Agrogenomics, biodiversity studies, model organisms, all of these benefit from having very high accuracy long reads that can be used to understand the composition of their genomes as well. And finally, most recently, I wanted to highlight a paper that was published by the group that's working on all of us being the US Population Sequencing Project, where I'm paraphrasing here, and you guys can go back and call me out on the more accurate statements, but small variants compared to HiFi long reads, HiFi data achieved the best F-score, the most reliable coverage of small variants as well as structural variants. They looked at medically relevant genes and say HiFi outperformed the other technologies in both precision and recall in medically relevant, challenging medically relevant genes, those dark regions of the genome. And essentially said at one point in the study, we should continue developing population scale cohorts sequenced with long reads only. We don't need to do more short read sequencing. There is certainly a place for short read sequencing in the world, but in terms of population-based studies and trying to understand the overall complexity and diversity of the human genome, we think we can do it with just HiFi and with just long reads, which is really an amazing thing to hear. Okay, so I want to shift gears a little bit. That's talking a lot about looking at genomes and assembling them, putting them into pan-genomes, understanding the entire genome structure itself and the complexity of all of the genomes that we all have together. There are other ways that you can use genome technologies, and one of those is trying to understand and end diagnostic odysseys for undiagnosed disease. I want to point out that our tools are for research use only. There are research projects that look at patients. One of those is an example of Genome Answers. What I love about this is the title of the paper is Genome Answers for Children, but the title of the project and the tool is Genome Answers for Kids. It's GA4K. I don't know if they thought children was a little bit fancier or more formal and thus better title for the paper, but GA4K is what you want to look at if you have any interest in this project. These are some 1,400 to 1,500 pediatric rare disease genome cases that had been fully sequenced with short reads. They'd been sequenced and analyzed. Every attempt had been made to understand what was happening with these kids using short read genomes. They took all of those genomes that had not yielded answers and used HiFi to look at them instead. And they got about 13% new explanations just by looking at structural variant integration. So just structural variants that weren't called with short reads were able to solve an additional 13% of the cases that had already been looked at and failed using short reads. You can see that there are also additional single nucleotide variants that are called. There's phasing information that's able to be found here. There's a wealth of information that's available from the long reads that can't be found with the short reads. That's really important to looking at rare and undiagnosed disease. I wanted to share a publication here as well from the group at Nationwide Children's Hospital in Ohio. So here they were looking at complex structural variation of tumor states in childhood tumors. Childhood tumors behave generally much more like inherited diseases than they do like germline somatic mutations, pardon me. So they don't, we just heard a speaker the other day at a conference I was at say, pediatric cancers don't act like adult cancers. Kids aren't small adults and children's cancers aren't small adult versions of adult cancers. They behave much more like inherited diseases. Here's a great example of an extremely complex rearrangement. Cancer is a disease of the genome, an extremely complex rearrangement that was able to be accurately understood and mapped using HiFi long reads that failed analysis with short reads, again, at this group at Nationwide. Here's a really interesting example of looking again at those medically relevant genes. If anybody is looking in areas of segmental duplication, if you're looking at regions that have medical relevance that are difficult to map, there's a big list of genes over there on the green box. These are sort of sometimes called NGS dead zones. Essentially what's happening here is somewhere in the evolutionary history of the organism, humans in this case, these genes have become duplicated. So that entire part of the genome was duplicated. That freed up, evolutionarily speaking, one of those regions to evolve away from the original function of that gene. So it alleviated the evolutionary pressure of retaining the function and the structure of that gene. What that means is that now you have two parts of the genome, or maybe 3 or 4 or 5 or 20 parts of the genome that are extremely homologous to one another, over 90% identical to one another in their sequence composition. When you take a bunch of small reads and you try to map small reads to a genome that has multiple copies of the same region spread across it, it becomes impossible. And what actually happens in many cases is the most valuable reads that you have are simply thrown out because they are unmappable. I don't know if they go in spot A or spot B, and so I cannot assign them to either, and I simply throw them away. In having long reads, we get rid of that problem. It's like solving a jigsaw puzzle that has very large and irregularly shaped pieces versus solving a jigsaw puzzle that has very small and almost interchangeable pieces. The former situation is far easier from a solving capabilities perspective. This work was done by this amazing group that's primarily at CHOP, Harvard, and Brigham and Women's, and feeds directly into efforts by the Genome in a Bottle Consortium, where they're building a database of complex medically relevant autosomal genes, including all of those that are listed over here on the right, and saying this is how you can use long read technologies to start to dive into these regions and make calls and make increase the understanding of what's happening for the cases where these are being adversely affected. Repeat expansions are another type of variant, of genomic variant, which has historically been really difficult to understand. Again, if you think about using short reads, if you have reads that are completely encapsulated within a tandem repeat, and that tandem repeat shows up not only multiple times within a gene, but multiple times within the genome, you have no idea where to put those reads. And so you're simply left saying, I don't know how many times this particular tandem repeat has shown up. With longer reads, we can span through and across those reads, those regions that have those variable numbers of tandem repeats. And we can start building a more comprehensive analysis of how those regions are being affected. This work was done at University of Washington. And in fact, all of the description of the number of repeats that are shown over here in this figure on the right, all of this was done in a single Sequel II SMRT Cell using an amplicon approach. So even the Sequel platform was very powerful in terms of looking at single genes. We know that we have ways that we can approach that in a completely different way moving forward with the Revio as well. There's also this really interesting piece over here where they look at the evolutionary implications of changing the number of repeats that are here. So these spikes right here that are all the same size show that this particular repeat shows up 6 times in the quote unquote normal human genome, and that it is evolutionarily conserved to have a different number of those repeats in other genomes throughout the clade. I'll leave that for you guys to dive into more deeply, but I think it's super cool. Another class of genes that are very similar to and in many ways represented within that dark region's genome are pharmacogenomics genes. Pharmacogenomics genes are those that are involved in metabolism and distribution of drugs and have implications for individual health in terms of medicine. Typically, these are genes that are expressed in the liver, and the cytochrome P450 genes are the biggest superfamily of genes that are largely involved with this. Because those genes and the CYP genes in particular are so similar to one another, it can be difficult to analyze those using short reads alone. So here we're showing an example of a pharmacogenomics panel, an enrichment panel that we've built with our partners at Twist that allows us to look at just those regions of the genome that are involved in pharmacogenomics. And you can see that we actually have phase blocks. The green and the purple up there are HiFi reads that read across the entire region spanning CYP2D6 and CYP2D7. 2 of the most important genes that are difficult to map reads into from a short read pharmacogenomics perspective. The IGV plot down here at the bottom is actually showing what short reads look like. And this piece right here, where there's a big island of non-mappable reads, is largely what makes it difficult to assign those reads to either CYP2D6 or CYP2D7. I wanted to share an example of why 5-base sequencing and the methylation status matters from a genomics perspective as well. This is work that was done by Matt Bainbridge at Rady Children's Hospital. Some of you may have heard of Prader-Willi syndrome. This is essentially, as we can see here, a syndrome in which uniparental disomy has occurred. And for the NDN gene shown here, the individual at the top has 2 copies of this gene, but they're both derived from the maternal allele. So I got both copies from mom in this case. This is an imprinted gene where the maternally derived allele is hypermethylated, meaning it has more methylation, meaning it is closed to transcription. So while this person has 2 copies of this gene that may have no deleterious mutations in them at all, they're not producing the functional NDN protein or the protein product from the NDN gene. allowing them to avoid the fate of Prader-Willi syndrome. All of the control samples shown down here have one hypermethylated copy from mom and one hypomethylated copy from dad, and are able to express that gene and avoid Prader-Willi syndrome. A similar example from work done internally at PacBio was looking at DIRAS3. And rather than that gene being important in and of itself, we're showing here that we can use the hyper and hypomethylation status. In this case, it's the paternally derived allele that is hypermethylated and the maternally derived allele that is hypomethylated as a variant in and of itself. That's important because it means that we can phase through that region of the genome. And if there are two flanking genes that we want to understand how they relate to one another in terms of their cis and trans status on both sides of this region, we can use this as parental imprinting as a variant type in and of itself and use the methylation status to phase across that part of the genome. Here's FiberSeq. This is the example that I was alluding to earlier, where we're able to look at the kinetic information to understand what's going on in the genome outside of just the CPG context. So the way this is work that was originally done by the STAM lab at University of Washington and has been adopted by other labs at UCSF and around the world. The idea here is we would like to know the chromatin modeling structure of individual single chromatin strands. So within all of the cells, within each strand, how are the DNA molecules wrapping themselves around chromatin? This is ideologically similar to something like a footprinting assay, where basically you use a methyltransferase and you purposefully methylate all of the exposed A residues. So you put a 6-methyl-A, you methylate A in all of the exposed residues, and you prevent methylation by steric hindrance from the histone itself for any non-exposed A residues. So now at a single strand, at a single fiber basis, you can ask which nucleotides are wrapped around the histone and which nucleotides are exposed to my methyltransferase and therefore accessible to other enzymes and genomic manipulation as well. This is really interesting work for anybody who's interested in the architecture of the genome at that level of epigenetics. And as I mentioned, has been used by multiple groups, including those at UCSF, to ask, even in complex mammalian systems, how do we see the modulation of individual chromatin structure happening in cells with different biological implications? I spend a ton of time talking about the human genome because I'm human, and we are anthropocentric organisms in general. But there are incredible projects that are looking at assembling complete genomes from complex metagenome samples using what we call MAGs, or metagenome assembled genomes. So I have a complex soil sample or skin sample or other biological sample. I have a host of different microbial organisms that are all present in that sample. By doing simply whole genome sequencing of all of the microbes simultaneously in a shotgun manner, let's just sequence all of everything that's in there, and then using high quality long reads to assemble them back together into complete circularized genomes, we can get some really amazing results. I would invite you guys to dig into this paper if you have any interest in understanding the genome complexity of very diverse microbial samples. This is really interesting work that can be done on the sequel, and I think will only be expanded further by the power of the Revio system. And then finally, the last thing I wanted to touch on today was, rather than looking at the genome, what about using long reads in a transcriptomic perspective? So a lot of the work that's been done on the transcriptome historically has been gene expression profiling. Let me take one little piece of the gene that we know is conserved, and I'll use that to count how many times that gene was expressed in a cell and what happened if I modulated the conditions that that cell was grown in. If it was a different tissue type, or if it was a drugged and non-drugged condition, starved or non-starved condition, et cetera, et cetera. What we now know from work by this team in Singapore, as well as many others, is that looking simply at the level of the gene and counting how many times a gene was transcribed or not is frequently insufficient to understand the true biology that's happening within samples. So here's a really interesting example of a gastric cancer sample where this team looked at two different isoforms, the isoform being the full-length transcript that's present for a given gene, not just a tag off of one end, but the full-length transcript. And we're actually able to discover that there were two promoters that were used in the system. There was the known canonical promoter, as well as a novel promoter that hadn't been previously described because everybody had always been looking at the other end of the gene and simply counting how many times it was there. When you do that gene expression profiling work and you look at the known promoter and you look at a Kaplan-Meier survival curve, this one looks weird to me, but the statistics actually say that if you're using the known promoter, it doesn't matter if you're a high expressor or a low expressor. Statistically speaking, your survivability is likely to be the same. Doesn't matter. This gene doesn't matter in terms of its expression profile. However, if you start looking at the novel promoter and you are a high expressor of the novel promoter, statistically speaking, your survivability drops dramatically versus if you are a low expressor of the novel promoter. So what matters here is not simply how many times this ARID1A gene is being expressed, it's which version, which isoform of that gene is being expressed. And that has direct implications for the biology that's present in this particular example. This group goes on to furthermore discuss a number of different alternative splicing, alternative stop, and other isoform variation classes that they can see in this particular model as well as others, and describe the full variability that's available within the transcriptome. Now, obviously, that's really interesting, but anytime anybody in the last 5 to 7 years said transcriptome, somebody immediately said, can I do it in single cells? Can I look at the single cell level? And it's a valid question because a lot of the biology that's occurring is actually occurring in the level of individual single cells. We want to be able to understand what's happening within the transcriptome to make one cell different from the cells around it. If you think about what I've talked with us about so far today, we have a system that's very good at reading about 15 KB worth of data at a time. And it doesn't have a ton of reads. The Revio will have a lot more reads than the sequel, but it doesn't have a ton of reads, about 100 million on the top end. And they're ideally about 15 to 16 KB. When we think about the transcriptome, there was a very clever group of folks at the Broad, and then actually some other folks who sort of through convergent evolution or convergent development, I guess I should say, asked the same question and said, what if we could just stitch a bunch of those circa 1 KB on average transcripts together and read the whole thing as one big read? So what if I took 16 of those isoforms, lined them up together, made one 16 KB super isoform, read the whole thing, optimizing the read lengths of the system, of the PacBio system, and then deconvoluted all that back out and got a bunch of isoform level single cell information. That technique has now been characterized as MOS-seq. This was the original preprint for the MOS-seq protocol. It works on single cells using the Chromium system from 10x. The reason that system is used predominantly is A, because there are a lot of them in the world and they tend to work very, very well. And B, they introduce highly conserved molecular hooks. So ways that we can use PCR and other techniques to selectively grab the isoforms that are present in those single cell samples that aren't necessarily highly conserved in things like bulk isoseq and other protocols, at least not yet. So this is a way we can concatenate or concatenarize. I don't know which is the right verb form there. We can stitch together a bunch of circa 1 KB individual isoforms into a 15 or 16 mer and read all of those simultaneously and start getting single cell data at an isoform level. I had a longer deep dive into this particular example, but I think I might truncate after just a couple of these, or maybe I'll move through them really quickly. This is my favorite example currently of using a MOS-seq-like concatenation approach to look at single cell biology at a transcriptomic level. This is work that was done in Switzerland, primarily in and around Basel, where they looked at ovarian cancer samples, a healthy section and then a non-healthy section across 5 samples. This was about 3,000 single cells sequenced at about 12,000 reads per cell. So this is the largest long read only single cell experiment that's been published to date. And they found some really remarkable things. So first and foremost, if you use short read sequencing and long read sequencing, and on the same samples in parallel, and you just do clustering, the screen stops working and you can't really speak to the data that's produced anymore. And then it comes back and everything's great. You actually get remarkably similar results. So if you kind of think about twisting this image through 180 degrees, the clustering pattern for both the long read and the short reads are incredibly similar to one another. So what this suggests is that you are getting very similar, if not the same results of overall clustering from a single cell perspective using either short reads or long reads. That's cool, but it doesn't really matter. Why wouldn't I just use the short reads? The short reads are cheaper anyway. Yes, the short reads are cheaper, but you're only looking again at one end, usually the 3' end of the transcript when you're using a short read sequencing approach. What that means is that you can't find any of the other isoform classes that are present that they talk about in this experiment. And what's really amazing over here are these 2 classes that represent 30% of the total isoforms that are described in this paper that were previously non-described in GenCode. They didn't even exist in the reference database. They weren't there. These are completely novel discoveries that are either new to the catalog or new and not described as being present in the cell types that they were detected in. So there's a ton of discovery space waiting for those that are going to move away from gene expression profiling and towards isoform sequencing for complex biology like tumors and oncology. I'm going to leave this for you guys to look at a little bit more. There are individual genes that they dive into here, IGF-1 being a great example, where they say, if we look at just the short reads, the clustering doesn't really tell us anything about that. But if we start looking at longer reads, we can see a class shift where all of the cancer cells express one isoform, and only a small subset of the non-cancer cells express that same isoform. And when they express that isoform, they appear to be in a transitional state. This is looking only at one gene. We can furthermore look at that and say, are we actually seeing cell transition events even based on just clustering? What if we start looking across more and more genes and look at other examples of where what used to be a more epithelial cell may be transitioning into a more mesenchymal state, which we know is canonically a hallmark of cancer and metastasis in particular. We can use the single cell data to look for somatic mutation. So when we oftentimes think about cancer, we're thinking about accumulating mutations, distinct mutations in individual cells or individual genomes. We can look at individual single cells, and here you can see the mapping of somatic mutation events, P53 in particular, in these cell types. And sure enough, all of the cells that are exhibiting somatic mutation cluster in the cancer cell type that was called by the clustering algorithms. Finally, what is probably my favorite example from this entire paper, the entirety of which I love, is the detection of this novel fusion that was completely missed by short reads. And so where I think that's the most impressive is over here. So in the short read data, when you look at that highlighted section, you can see that there are yellow cells that are popping up, where the short read data said We think that there is both TESPA1 and IGF-28P2 expression, co-expression simultaneously in those cells. We see hallmarks of both of those two things. And in the long read data, what you can see is actually true is that no, those are not cells that are expressing those two genes that have never been seen expressed before together. They're actually expressing a novel fusion transcript that had not previously been described. And so you're not seeing expression of two genes, you're seeing expression of one novel fusion transcript. And all of those yellow co-expression examples drop out of that data set. It's a really stark example of understanding more deeply the biology that's actually occurring in these samples. This was the author, this was a statement by the author where they essentially said, paraphrasing here again, so forgive me. We're here demonstrating that long read sequencing provides a more complete picture of what's happening in the samples, and we don't really think we need to do short read sequencing just to look for neoantigens or for other interesting biology occurring in these models. So we're right at the end. I'm going to jump through all this stuff and then ask if anybody's been holding on to questions. I have a couple of requests. Anybody who I haven't met before who wants to meet me and chat with me and thinks that this nerdy stuff that I love is as interesting as I do and chat some other time, please come find me. I have a tiny little briefcase that holds my business cards. It is my favorite thing that I own from a professional perspective in the world. I would love to undo the clasps and hand you a business card. It's really fun for me. Tells you something about how I have fun. If I've made any claims that you don't believe, Roger's going to call me out on this for sure. Challenge me on it, and let's find ways that we can understand where the misunderstanding is and dig into trying to solve some of those issues together. The next time you're thinking about doing genomics work of any kind, think about using PacBio. If I was going to guess, you're probably thinking about doing it on Illumina and using short reads. That is sort of the dominant paradigm in the genomics world. At least think about if there are opportunities to use long reads, to use HiFi, and to perhaps discover novel biology that's really important for your system. I'm always here for consultation. I put Molly's information up as well because we would love for you guys to continue working really closely with this amazing team at the University of Wisconsin here in the Biotech Center and doing some really amazing science. And finally, beyond all of that, I hope all of you take care of yourselves. I hope that you find ways to be well and that you take care of one another. Thank you very much. I really appreciate it. Molly, did you have a question on that? Was there one that came in? Oh, good. I thought you raised your hand. Cool. Well, we covered a ton of material. The back half in particular, we were flying through a survey of the ways that HiFi sequencing has been utilized in the literature. My goal with that was to simply expose people to the not even the full breadth, but a pretty good cross-section of the ways that HiFi sequencing can be used. If anybody wants to follow up, explore ideas, ask about something that's never been done before, I'm always around. I'm always happy to chat. So thanks for everybody for coming out in the snow and the sleet and the bad weather. And for those of you who joined us online, thanks for your time. I appreciate it. Oh, everybody make sure that you read this statement at the end about all of our trademarks and don't use them incorrectly, because I certainly didn't.